Well, welcome to the fourth webinar in our Introduction to Designing Adaptive Trials series. And today what we're going to look at is designing seamless phase 2-3 trials. We'll look at, actually just ignore that outline, I'm not sure it follows that. Let's just go straight in to seamless phase 2-3. It's a fairly simple idea. I'm actually expect that everyone watching this has actually got an idea of what a seamless phase 2-3 is, but just to be sure, it's a simple switch from the conventional drug development program where you do a phase 2, you make a go no-go decision, you possibly pick a dose or a treatment combination, perhaps a treatment population, and then you do some planning, and then you start 2 phase 3s in parallel. The idea of a seamless phase 2-3 is that you do a lot of your planning up front so that you can run seamlessly from the phase 2 into the phase 3, and you don't have any of this, what was called white space in between the phase 2 and phase 3. And our objective principally is to save time. We get our phase 3s finishing that much earlier. How much time this is obviously dependent, but it's rarely less than about 6 months, but often more between the end of phase 2 and the start of phase 3. So we've got this advantage of saving time. We can possibly also save phase 3 sample size if we can include phase 2 data in the phase 3 analysis. So the initial idea of a seamless phase 2-3 didn't get a terribly warm reception from the regulators. Their initial reaction was, you call it dead time or white space, we call it thinking time. And essentially the regulator is going to be asked to approve the phase 3 design, not on the basis of seeing the phase 2 data and saying, yeah, the phase 3 design, but they're going to have to prospectively prove it based on looking at the design of the seamless 2-3. They will be concerned typically about how you're protecting the integrity of the trial, which is normally to use an independent or firewalled team to produce the analysis, the interim analysis report, and make the end of phase 2 decision. And they will also want to know what the basis for the dose selection is, if dose selection you are indeed doing at the end of the phase 2. Because this is going to have to be prospectively designed and the rules specified. The second complication is, what are we going to do about enrollment? Are we going to continue enrollment after phase 2 is fully enrolled? Or are we going to pause it until phase 2 is fully followed up? And if you've got quite a long endpoint, then that may mean quite a significant pause in the enrollment. Now, it might be that instead of using, it might be we have a very quick endpoint for phase 2 and phase 3, which is fine. It might be we use a different endpoint in phase 2. We use some early endpoint to make our decisions on, and a later endpoint in phase 3. It might be we've got a late, some time to the endpoint, but perhaps we can predict that late endpoint in some way, either from a different endpoint or from just early visits. And if we're going to pause enrollment and then try to resume it, what risks are we running? How easily would it be to restart enrollment? Might we have lost some sites who've gone on to recruit subjects into the similar patients, but into a different trial? One distinction in between seamless phase 2-3 designs is whether they are what's called inferentially seamless or operationally seamless. So operationally seamless, we simply do this. We do phase 2, we make the decision, and then we run our 2 phase 3s. The inferentially seamless is where we borrow the phase 2 data, particularly the arms that we keep from phase 2 into phase 3, and use that data in the phase 3 analysis. And initially, that was it was just kind of like a binary decision: is inferentially seamless or operationally seamless. But over time, it became apparent that actually one could do this inferentially seamless design in a number of ways. You could use all the data from the phase 2 in the phase 3. You could use just the arms that you carried forward from phase 2 into phase 3. Or you could take all the data on the treatment arms, the experimental arms in phase 2, and pool them and combine them on the one arm kept into phase 3. The idea here is that this mitigates or eliminates the effect of the multiplicity that you have of choosing between arms at the end of the phase 2. If you consider the null case and you're choosing between 2 or 3 arms, then what's going to happen is you will consistently take the arm with a better result. In the null case, that would be they just have a random high, and you'll get some multiplicity effect and have to take some small alpha adjustment to control the time and error at 3. But if you pool all the data onto the arm you select, then of course there's no selection bias going on. There's no a multiplicity effect, and you shouldn't need to have to pay a time and error adjustment. Perhaps we don't take the treatment data at all from phase 2, phase 3. Maybe we just take the control data, or maybe we just take the subjects who haven't completed yet. So we transition out of phase 2. We don't do a full follow-up. We just take a data snapshot, and we only use perhaps the complete subjects at that point, and there'll be a number of subjects we've already recruited but haven't finished, and maybe they can be used, or maybe they can be used if they haven't reached a certain point in the phase 3. And then there's some design aspects in the complications, which is how are we going to select the arms at the end, or arms in phase 2 to include in phase 3? How many arms are we going to select at the end of phase 2 to include in phase 3? And how do we size the phase 3? And of course, for the company, justification of doing this is quite easy. The time savings can be quite significant. A minimum of 6 months, easily up to a year. And then how do we justify this to regulators? After all, they're not so concerned about the advantages of saving time to the company. If there's high unmet medical need, though the saving of time can be a good argument for the regulators, because we're making available, we could be making available a treatment where there currently isn't one. They might also be persuaded if the phase 3 requires very large sample sizes, and so they can accept that this is a burden, a difficulty in developing drugs in this area, and they could be prepared to allow you to use the phase 2 data to somewhat reduce that phase 3 size. It might be that this is a very rare disease, and so even though perhaps the required sample size isn't large in absolute terms, it's large in terms of the patient population that's available, and doing a fully powered phase 3 might be difficult or very time consuming because recruitment's going to be very slow. And in this case, they might also be prepared to use the phase 2 data in phase 3. And one other area where they might be prepared to let you use phase 2 data in phase 3 and be prepared to do a seamless phase 2, phase 3 is where you've actually got almost the opposite of the previous case. You've got a very well understood disease and a very well understood endpoints. And so there's very little risk of unknown unknowns arising at this end of phase 2. So we're going to consider an example now, and we'll look at perhaps not all of those issues, but some design issues by working through this example in facts. So let's say we've got in phase 2, 3 arms, perhaps 3 different doses versus the standard of care, the control. Let's assume it's a continuous endpoint, which is available after 6 months of follow-up. And there's a standard deviation on that endpoint of about 2 points. Our expected improvement on the standard of care is let's say 1 point, and our target improvement for the treatment is 2 points, so a treatment effect of 1 point. And we're prepared to accept, because this is phase 2, quite a high false positive rate, high type 1 error of say 20%. And if we do a Bonferroni adjustment, then that means we're prepared to accept sort of 6.66% error, type 1 error on each arm. And if we require 90% power, then a conventional sample size calculation tells us that we're going to need 63 subjects per arm. If we then select one of those arms and take it to phase 3, and we require now, of course, a more conventional 0.025 single-sided alpha and 90% power, then we're going to need 85 per arm on the 2 arms that we take into phase 3, the standard of care and the 1 treatment arm. And let's consider just 4 scenarios for the sake of this example. Of course, we'll look at the null, where every arm has just this 1 point improvement over mean improvement over baseline, including the standard of care. Then we'll look at one where one arm, let's say it's dose 1, has all the effect. It has 2 point improvements, but there's no improvements on the other arms. So this is a kind of 1 effective arm and 2 null arms. Then we'll consider another one where we've got the full improvement on one arm, let's say dose 2, and we've got half that treatment effect on each of the other arms. So 1.5 and 1 point on dose 1 and 1.5 on dose 3. And then lastly, let's assume we've got some kind of linear dose effect. So we see a full 2 points improvement on dose 3 and 1.33 mean change from baseline on dose 1 and 1.67 mean change from baseline on dose 2. And what I'm going to look at is what is the benefit of using phase 2 data in phase 3, and what's the effect of doing that borrowing on type 1 error? How much do we have to adjust the final analysis in phase 3 to reduce the type 1 error inflation? And once we've done that, have we actually got any benefit? And to start with, we're actually going to ignore this 26 weeks to the final endpoint. We're going to come back to that and address that in the second part of the talk. But in the first part, we'll just assume the simplicity that it's a quick endpoint, and we'll ignore the overrunning issues. So I'm going to assume that all phase 2 subjects complete in phase 2, and then we do phase 3. And there's very little gap between the two because the follow-up is very short. So we're going to do this in a couple of stages. So the first is we'll create a FACS core design, which is just going to simulate the phase 2 part. And this, the advantage of this is that we can study and check the behavior of the phase 2 without also simulating the whole stage thing and having all the data around for phase 3. And it's much easier to concentrate on getting to phase 2. And then we'll take the resulting FACS core design and we'll import it into a FACS stage design, which is the FACS engine we use for where we're simulating both a phase 2 and a phase 3, whether they're separate or seamless. And then we'll simulate them both together. So let's get into that. I hope that's sufficiently visible. Perhaps we can change the display settings slightly. Okay, hopefully that's a bit better. So it's going to be a FACS core design with a continuous endpoint. To start with, we're not using any adaptation. In this example, I'm not going to use any longitudinal modeling. We're not going to simulate the baseline score. We're going to enroll subject continuously. Our maximum subject size is going to be 63 per arm with the 4 arms. So that's 252. And we're going to have a better responses improvement. And to start with, we're just going to leave the final endpoint at 1 week. We will have a control arm, standard of care. We'll have 3 treatment arms. I'm not going to change their names. And then we're going to have these 4 scenarios, like the null. There was arm 1. I called it peak at arm 2. And something like arm 3 linear. And all of them, I have to remember to set that the standard deviation of the response is 2 points. And in the null, we have a mean change of baseline of 1 point on all arms. And arm 1 is successful. It's a response of 1 on control and doses 2 and 3, and a response of 2 on dose 1. And I'm going to mark that that is the arm that should succeed. So when FACS has done the simulation results and looks at which arm has been picked, it can then tell me what proportion of times the arm I want picked, or one of the several arms, if I check multiple of these, when was that actually one of the selected arms. Now I'm going to have this peak at arm 2 with half the response on each of the other arms. So here now, it's going to be much harder for the trial to determine that dose 2 is the best arm, because dose 1 and dose 3 have now got some kind of effect size. Whereas in this scenario, we should be able to pick the correct dose most of the time. And then lastly, in the linear case, the same thing, and now we've got also some chance that, particularly dose 2, the data will look a bit better than dose 3 if we're unlucky and get some random highs on dose 2 and random lows on dose 3. So one of the things we're going to also want to study in our phase 2 is how good it is at dose selection, not just its power and type 1 error. At this point, it doesn't really, because I'm not really considering time, it doesn't matter what the accrual rate is, but I'm going to set it now because this file will get copied and modified to create the other designs. And I'm going to set the simple flat mean accrual rate of six per week. So that gives us an accrual time for phase 2 of just under a year. We're not going to consider dropouts in this design. And now we get to look at what are the quantities of interest where we're going to be looking at. And here I'm going to keep it very simple. So dose selection is just going to be the dose of the maximum response. So I wanted to look at what arm has the greatest probability of having the maximum response, and we don't need a minimum effective dose or an ED50. I'm not interested in beating control by some delta. I'm simply going to be interested in beating control. I'm not going to be looking at predicted probability of future trials, but I am going to use a p-value. It doesn't matter what I do with missingness because I'm not going to simulate any missing patients, and I'm going to use an unadjusted p-value. So at this point, my decision quantity is going to be the p-value of the dose with the which has the greatest chance of being having the maximum response, which of course is pretty much the same thing as the p-value with the arm with the smallest p-value. And on the design, I will set this up for later use where I use a Bayesian analysis, and I'm going to have a simple prior expectation of a response of 1 point, which is what we expect on control. And I'll set the standard deviation to perhaps 4, which is twice the standard deviation of the endpoint. So the weight of the prior is equivalent to 1/4 of an observation. And on the estimation of the sigma, I will center it in the right place. I will say the central value of the prior will be 2, because we're going to simulate standard deviation of 2, and that's our expected standard deviation, but I'll only give it a weight of 1, so it's a very diffuse prior. I don't want any additional frequentist analysis. I'll use simple one-to-one allocation. We might be able to improve the power by doing a kind of a root k to 1 allocation, but we're not going to look at that today. And then we're just going to look at simple futility and success criteria. And we just got the p-value, and I said what I was going to have an overall type 1 error of 0.2, on Ferroni adjusted for my 3 arms. So my limit, my alpha level will be 0.067 or 0.066. And I'll set the corresponding threshold for futility. And we'll just run 1,000 simulations of each. And you can see that zips through them pretty quickly. You can see our probability of late success is around 16%. Our power is about 90%, 93%, 94%. And right down the other end, we have our proportion of trials where we were successful and selected the correct arm, where the correct arm is defined by what I checked here in the scenarios. And the selection, actually, currently, selection is in a slightly odd place. We're moving this to a more logical place, but it currently lives on the variance tab. This is so this is saying the dose will be selected by being the dose with the highest probability of being the max. So these results look pretty reasonable. I've got my time and error controlled. I've got my 90% power, and we're losing a little bit. We've got about except where the other arms are null, we've got some like 10 to 15% chance of picking a second best arm. But we probably can't do much better than that without increasing the phase 2 sample size. And we can, for the sake of time, I only ran 1,000 simulations, but somewhere in all these facts files I've got open, here I've got one where I ran 10,000 simulations. It doesn't take a lot longer than the 1,000 we've just seen, but we can have a lot more certainty about these operating characteristic estimates. And it's these numbers from running 10,000 simulations that I'll be using in the slides. So what we want to do now is turn that, so we've got our phase 2, its operating characteristics look reasonable, so we're ready to simulate the combined phase 2 and phase 3. So I want a new facts file. It'll be a staged design, and I'm going to create from this existing core design project. So it asks me to find the file to create it from. So I just called it temp. It says, well, it's removed final success criteria to allow the trial to graduate instead. And now it's prompting me, what do I want to call this? So I'll call it temp staged. So you can see the setup is as we already had it. And I think, but we've now got 2 design tabs, stage 1 and stage 2. The stage 1 is as I specified it for the core, but there's this slight difference on the success futility tab is now success futility graduation. And what we've done is specify that there's now three possible outcomes at the end of phase 2. There is futility, which means we won't go any further. There's graduation, which means we will go to phase 3. And there is success. And this means some kind of super success. The trial was so good that actually we don't need a phase 3. And so, again, we don't go to phase 3, but we declare overall success without it very early. Obviously, that might be quite a rare circumstance that can actually be used. But what you do, naturally, the super success criteria would be very high, and then you've got the futility criteria. And so the default, if you haven't met either of those, is to graduate. So we don't specify graduation criteria. It's if you haven't met the futility or the super success. So we're not setting super success. We always have to run a phase 3 if we want to be successful. So we just retain the futility criteria. I'll come back to the transition tab in a moment. The stage 2 design, the tabs are the same as we have for phase 1, the stage 1 design. And in fact, I could even borrow directly from the stage one design by checking the mirroring. Again, I don't want any sort of separate frequencies to output. We don't need that. Allocation is different. Allocation allows me to specify the allocation ratios depending on the number of treatments that have been taken into the second stage. Here at the moment, because I haven't set up any transition rules, the only possibility is me taking one treatment arm interface into the second stage, so there's just one row in the table, and it's set to one-to-one allocation. So let's go back. And then the success futility criteria. So again, we're going to use the p-value, but now we'll start by just setting it at 0.025. And now, before we go on, we need to look at the transition tab. So the transition tab has got two parts, the dose selection and the data inclusion tabs. The dose selection does what it says. It allows us to specify the rules for how we're going to select doses from the first stage into the second. There's two ways of doing it, what's called the standard selection logic and the representative arm logic. The representative arm logic is for a situation where you've got multiple arms in the first stage and you want to possibly take different groupings of those arms into the second stage. It might be, for instance, that you've got arms which are different combinations at different doses of different treatments. And it might be that, well, if you take the lowest dose of drug one, then you always want to take its combinations with the other doses of the second treatment. So you make your decision just based on the lowest dose, perhaps on its own. And if that's taken to phase 3, then you're also going to take its corresponding combinations. So it's for that kind of decision making. You might be simulating actually not so much a phase 2, phase 3, but a sort of phase 2A, phase 2B trial. We're going to use the standard selection logic, and that's got 3 ways of setting up rules, and basically they're all combined. I can set a rule per dose. So I would pick the dose and then I'd set what the rules were for keeping that dose into the second stage. I can do a target dose decision. So this is possibly the simplest. And it basically, I can keep the best, i.e. the dose with the highest probability of being the max. And that's actually the rule I want. It's very simple. And then there's an all dose decision, which I don't need, but it allows me to specify a minimum number and a maximum number, and then what QOI to use to sort them, and also to specify other filtering rules to rule doses in or out. And then separately, I get to say whether we keep the control or, if we had one, the active comparator. And then if that's a little complicated, we can look at the summary where it'll just summarize what the rules are that I've set. So again, we're ready to run some simulations. And these will take a little longer than just doing the phase 2 or the stage 1 by itself, of course, because it's going to do the second stage as well. And basically what FACS is doing is it's running the FACS simulators twice. It runs the FACS simulators to do the first stage, and they output all the information that FACS will then need to run the second stage. So what happened in the first stage, what was the data, what was the conclusions, and then it runs FACS again to do the second stage, and it will read those in and then carry on with the rest of the simulation for the second stage. So The two stages are very simple. There's no interims. There's only one endpoint. There's no longitudinal modeling. So as you can see there, they're being simulated pretty quickly. And again, it's really only a few minutes to run something like 10,000 simulations of each scenario and get reasonably good estimates for the operating characteristics. And I'm just doing it only 1,000 times here just to keep the time down during the training. While we're doing that, let's just go back to the slides. So we've looked at success and the probability of selecting the best arm. And what we're going to do now is, so this initial design, we've only, I didn't, no, there's something I forgot to do. Let's cancel that. It takes a little while while it cleans up all those files it's made and stops the processes. What I forgot to do was on the transition tab. There we go. It'll come back to me eventually. On the transition tab, I forgot to hit the data inclusion options. Oh, I've been clicking ahead too much. Data inclusion options, and I had as a default, perhaps incorrectly, is included in full, and at this point I wanted the not used option. So rather than continue running those, I will jump to the ones I've baked already. Here we go. This, I believe, is separate. So we're not using the phase 2 data in phase 3. And now we can look at the overall success. So this is, we're only successful now if we've been successful in the phase 2 and gone to phase 3, and then the phase 3 has been successful. You can see the combined overall Teichmann error is very small, because we've got alpha level of 0.025 in the phase 3, plus we set the Teichmann error level at phase 2 to be about 20%. So 0.2 times 0.025 is 0.005, and the difference between 0.35 and 0.005 is actually within simulation error, even at 10,000 simulations. And then you can see that our power is now around about 80%. So basically, and again, that's kind of what we should expect. We had about 90% power in phase 2. We've got about 90% power in phase 3. So 0.9 times 0.9, we've got about 0.81. And interestingly, it's somewhat less in these two scenarios. So this first scenario is where just one arm has the full treatment effect, the other two are null. So we've got a very, very high probability of selecting the right arm. In fact, we can see that. So we've got a number of the graphs are now grouped depending on what you want to look at. There's the stage 1 graphs, there's the stage 1 dose selection graphs, the stage 2 graphs, and the overall graphs, which won't be that interesting at the moment. But we can look at the dose selection graphs, and we look at the proportion times each arm selected, and you can see here that the dose 1 is selected almost 100%, and the nulls of dose 2 and dose 3 or almost zero. So you can see that we're in this instance where we can be really only the type 1 error control in the overall null scenario is the important one. The type 1 error control here is kind of easily achieved because we almost never take a null arm into phase 3. But here, where the other arms have got some kind of treatment effect, we do take those arms with their treatment effect of 1.5 some of the time. It looks like about perhaps 0.5 each, so perhaps 10% of the time we haven't taken those two through. And of course, those don't have the full treatment effect, so that the power of the phase 3 is reduced. So we've got this slight drop in the overall performance because of those occasions, and similarly in the third scenario. And we can see the proportion of times here. We can see the proportion of times an incorrect arm is included in a successful trial. So even when we've taken the incorrect arm, because they do have some treatment effect, they are successful some of the time. And now what we can do is say, well, what would be the effect of including phase 2 data in phase 3? So here we have, without making any other changes, we've simply gone to the transition tab and included the phase 2 data when it's on arms that are kept into the second stage, into phase 3. And we can see now that our type 1 error is, let's just show you, so the final, nothing else has changed. So the final success criterion phase 3 is still 0.025, but because of this multiplicity effect at the end of stage 2 and the dose selection, we're always picking the arm with the best data. So we're always picking the arm with a random high to pull into phase 3. And when we include that data as well in phase 3, it's causing this type 1 error inflation. And of course, it's inflating the type 1 error hugely compared to not including the data at all, where it's 0.0035, but it's also taking it over 0.025. And of course, we're getting a little more power at the same time, because that too is being, the power is being inflated. In that case, not just because of the multiplicity, but because of the additional data on both the treatment arm and on the control arm. There's an interesting question on what the regulators would require here in terms of adjusting the type 1 error. And I don't have an answer. I'm not sure what the requirement is. And in fact, it may well be that the regulators might be inconsistent in those questions. Oh, Kurt, I see we have some questions. Is there anything I should be addressing, or are you addressing those by typing into the Q&A?
I have been typing furiously, so I think I got them for the moment.
Thanks very much. So we've got sort of two possible strategies. One is, can we pull the type 1 error down so that we bring this overall error rate down to 0.025, or do we bring the alpha level down so that we bring the type 1 error down conditional on going to the phase 3. There are some tools to help us normally, in fact, to help us with this. Turns out they didn't work so well in the staged situation. So I'm going to do it manually. So what I did was, let's find the simulations file. So we get quite a lot of output in stage design. We have the simulations file for stage 1. So this is the result of all the stage 1 simulations at the end of stage 1. And we get a stage 1 summary file. And we can also have the weeks files for stage 1, which would include any interims that had occurred. We haven't got any here because we're not being adaptive yet. There's also a dose selection summary, which is slightly different from the main summary. I'll explain these two as well. There's also what's called a CD, simulations and summary. If you're doing a seamless trial and you transition from stage 2 to stage 3 with some of your stage 2 subjects incomplete, you can ask, and the default is to have it output, which is why it's here, even though we don't need it. You can ask for FACS to output a complete data, a stage 1 complete data summary and simulations. And what these have done is said, well, ignoring the stage 2 subjects that are coming in, if we just follow up the stage 1 subjects and then do an analysis when they're all complete, this is what the analysis would look like. So that's what these CD files are reporting. Anyhow, what I want is the simulations way up here at the end of stage 2. And if I pull them into a spreadsheet, I don't bother to have Microsoft Office in my virtual machine. That's out in my Mac environment. It's taking a little while to load because this is 10,000 simulations. So it's 10,000 rows in the spreadsheet. Each row is the result of one simulation. And there's lots of columns with all kinds of data in. Now the QOIs are all towards the end. And the one I'm interested in, of course, is the p-value. Whoa, I didn't mean to shrink it. It's this one here, column DL. And what you can see is we have a bunch of them where the result is minus 999. What that means is there is no value. That's FACS's number to represent there is no value here. And of course, what's happened is the second stage has not been run. So there is no value to be output for the second stage p-value. But if we look down, we'll see occasionally there are a few, and that's where in the null, we went from second stage and we went into the went from the first stage into the second stage. We had that's our 20% type 1 error in the end of the first stage. So if I sort all my rows based on that value, so we get all our p-values nicely sorted. So that's going to be 10,000 rows. Get there in a moment. nearly there. Now, of course, it's much better to do this in R, but if I show you it being done manually, it's much clearer what's going on, even though what I should really have is an R script to make it more reproducible. So we're going to go data, we're going to go sort, we're going to take the DL column, and I want them in descending order. So this is the largest p-value we saw, and what we can, and then what we can do, and then what I've set here is to summarize by counting when I select cells. So I can go down and I can select, so and I'm doing this the wrong way around, aren't I? That's what I really want to do, is to go to the boundary here. And then go up. So I want to say I don't want more than 250 successful trials out of my 10,000. That's 0.025. And there you can see that if I want to limit the number of successful trials to be, in total, to be not more than 250 out of my 10,000, I need to set my alpha level at about 0.02. If it's as a proportion, not of the total number of trials, but as the proportion of the trials that made it to phase 3, then it turns out I need to set the threshold at about 0.013. So we can run both those simulations. So here I've set the final alpha level to 0.02. And as you can see, that has indeed controlled the Teichmann error below 0.025. Obviously, yes, now this has been run with the same random number seed, so I'm pretty much guaranteed to have achieved that. Obviously, that level might be slightly wrong because of simulation error, so that's something one would need to resimulate and check on, and perhaps be a little more pessimistic about, to give ourselves a little buffer if we wanted to convince the regulators we had truly controlled Teichmann error below 0.025. But if the regulators actually want us to be even more cautious than that and have Teichmann error controlled conditional on going to phase 3, then we set that Teichmann error, that alpha level to 0.013. Here on the success futility tab. And something quite odd, I think it's quite an odd result, is that, well, as we would expect, this reduces our overall probability of success to below 0.025. It's about 0.018. but the effect on our power is very, very small. We still essentially got 90% power in this scenario. We dropped from 91% to 90% here, and we dropped from 92% to 91% here. So although we've dropped this alpha level, what feels like quite a lot, from 0.02 to 0.013, actually the impact on our power is almost negligible. Oh, there's one other option to look at, and that's pooling all the data, pulling all the treatment arm data. There we go, pooled. And that has, oddly enough, it's pulled the alpha. We've got an alpha level at the same as when we had 0.013 and used just the data on the right arm. We've still got 90 and 91% power in these two alternate cases, but in the case where we've got two null arms, all that null data being pulled into the pool has pulled down our power quite a lot, from what would have been 90% to only 70%. So if that is genuinely thought to be a possible alternate scenario, probably that might make using the pooling approach unattractive. So this is where we've adjusted the time and error. Oh, and the last thing to note is, of course, this really hasn't affected the ability to select the right arm, because that's essentially dependent simply on the first stage, which we haven't changed. So if the regulator's happy to allow stage 1 data, the phase 2 data, to be incorporated in the phase 3 analysis, it looks like paying the penalty of adjusting the alpha to reduce to offset the multiplicity in the first stage is, well, it's still worth doing. Actually, we can also do something similar in the frequentist domain. Tools like AdPlan, and I imagine East, and R packages like RPact, they allow us to look at and simulate group sequential designs with multiple arms and arm selection. So using, in this case, AdPlan with test of means multiple arm, we've set up just 2 stages. I'm not doing group sequential anywhere else. I'm just doing the interim at the phase 2, phase 3 boundary. I've got 3 arms. What it's going to do is, rather than compute a p-value overall and do this kind of alpha adjustment that I did via simulation, it's going to control the type 1 error analytically by using p-value combination. So it's going to take the p-value from the phase 2, p-value from the separate phase 3, but then it's going to do a combination using an inverse normal to give us the final p-value for the overall. And we're going to do a simple Bonferroni for the p-value at the end of the first stage. I've specified the boundaries using alpha and beta spending, and I've manually set the alpha spending at the first stage to be 0, because I don't want a success at that point. That's the end of the phase 2. And then I've set the beta spending to be 0.1 at the end of the first stage, the phase 2, and then 0.2 overall. So that's our kind of 90% power in at each stage. And then I've simply simulated. So in our plan, we don't, in this setting, we don't get to specify the effect on control, only the treatment difference. So here I've just looked at that first alternate scenario, 100, standard deviation of 2, and I've said simply select the best arm. And then I've specified the sample sizes as 63 on each arm in the stage 2 and 85 on each arm in phase 3. And then sure enough, we get these results here, where it's got the probability of rejecting arm 1, a successful arm of 0.895, which lo and behold is exactly the same power pretty much as we get in the from our simulation adjusted overall type 1 error. And similarly, their type 1 error in the null, which was this second line of simulations here, is 0.017, which again is exactly the same as we're seeing. So the that's in the 013, not pooled, right? So we're finished with you. Yeah, this one here, 0.017, 0.018. So very similar results. So the next thing I want to look at is can we still do seamless phase 2, phase 3 if we have a serious delay in the endpoint? Now, there may be ways of doing this by using longitudinal modeling or some early endpoint. Obviously, the effectiveness of that gets down to, well, how much do you believe your expected correlation between the early endpoint and the late endpoint? So I'm not going to look at that. I'm simply going to look at the 26 endpoint, and I'm going to see if we can still come up with some effective seamless design. Just to note that if I simulate a 6-month delay between the end of phase 2 and the start of phase 3, then the average duration we're seeing in the simulations are 80 weeks in the null, which is pretty much mainly dominated because the duration is dominated by just the first stage, the phase 2, where 80% of the failures occur. And in the alternate scenarios, it's about 140 to 143 weeks. So it's just a little bit shy of 3 years doing the phase 2 and the phase 3. So what we're going to do is we'll make the phase 2 adaptive. We'll add an interim when 252 subjects are not complete, are enrolled, and then force the trial to stop. Because this is the point at which, if you were keeping the same sample sizes, we would now want to be starting to go to phase 3. But of course, we're going to have an awful lot less information. At the point where 252 subjects are enrolled, actually, I'm not presenting here, so I can actually make this edit. The subjects accrued in the last 26 weeks will still not have completed. We're simulating, on average, 6 subjects a week, so that's nearly 150 subjects. So in fact, if we do this, we'll actually have only complete information on 100 subjects instead of 252. It's probably not loaded up. But I don't think I need you anymore. So let's open. Stop at 252. So I've made the designer adaptive. I've made sure I've got a 26-week endpoint. Everything else has been kept the same, but now we get to set up an interim. So I said there's an interim at 252 subjects enrolled. And at that point, I set up complementary success and futility rules here arbitrarily at sort of 0.8 to force the trial to stop. So all the simulations, unless I'm unlucky and actually get one where this probability is exactly 0.8, all the simulations will stop at the interim. And you can see that 0.8 was actually done with a little bit of tuning. Well, perhaps not quite. We've not got 90% power, and we've got almost 40% type 1 error. And if we look at the probability of selecting the right arm, that's got worse as well. So it's perhaps not, well, it's not unexpected because we've got this less information, but it's kind of putting numbers on just how bad things will be if we tried to transition to phase 2 to phase 3 at the 250 enrolled rather than 252 complete. So what if we allow phase 2 to recruit more subjects? So if we allow it to recruit an extra 156 subjects up to 408, then when we make 200, at that point, we would have, when the 408 subject is accrued, we'd have 252 enrolled. But we don't really want to go to 408 if we can avoid it. So let's make the first interim at 252 recruited and add interims every 8 weeks, which is roughly going to be 50 subjects roughly. We're going to allow adaptive allocation to give ourselves a bit more power and allow early stopping. And then what we do is to simulate, initially, we simulate this without any early stopping rules, and we output a weeks file for every simulation. So we output 1,000 weeks files. And then we use this wonderful graph, in fact, called the interim versus final scatter plot. And that looks like this. And what I've done here, I've only simulated the null and one of the alternates, just to keep the number of simulations down, because it takes it a while to draw this graph. The more simulations you've got. So that's also why I'm only using 1,000 rather than 10,000 simulations to do this. On these controls here, we can set what quantity of interest I'm using for the final decision, for the final decision and the interim decision. I set what thresholds I'm using for success and futility at the end and at the interim. It allows me to pick a particular interim, and then I redraw, and it plots this. And the red dots represent simulated files that, given these interim stopping rules, would have gone to the end. They wouldn't have stopped and would have been futile. The light pink are ones that would have stopped for futility and would have been futile at the end. The light blue are ones that would have stopped for futility but would have gone on to be successes. The greens are the ones that don't stop and go to the end to be enough successes. The light greens are ones that would stop at the interim and would have been successes at the end. And the purples are ones that would have stopped early for success, but actually, if we'd gone to the end, would have been futile. So you can see here, I've set an interim stopping threshold of 0.95, which is up here. So you can see we've got this band of dots, and the ones where the value at the final analysis is high enough, they're successes. But if they don't meet that, then they would have been futile at the end. So that's why we've got this band of pink representing the ones that were stopped early for success, but were actually futile at the end. So they're kind of like a type 1 error. And then we've got the stacked bar chart for each scenario. So you can see the stopping rule would have been great for stopping early in the alternate, but of course it's got this problem that we've got nearly 10% type 1 error, early type 1 error in the null case. So we probably want to make that futility, write that early success boundary up. And then we can repeat that at successive interims, because obviously at successive interims, the interim value gets closer and closer, more and more, better and better correlated with the final value. And we can sometimes start to move the thresholds to be more aggressive. In the shortness of time, I'm not going to actually show that in facts. You'll just have to take my word for it that that's what it looks like. And when you use it, you just have to you have to be a bit patient with the redrawing, because it can take a minute or two, even with only a couple of thousand simulations. If you've got even more simulations, it will take even longer to redraw. So looking at that, I've picked boundaries of 0.99. So the probability of the Bayesian posterior probability that the treatment effect on the dose with the maximum effect has to be better than control. That probability has to be greater than 0.99 to stop for success. The stopping for futility, I set it to that probably has to be less than 0.1 to stop at the first interim, but then I can relax it a bit to be less than 0.15 at the second interim and less than 0.2 at the third interim. The fourth interim is going to be 3 times my 8-week interval, so 24 weeks, and I've nearly enrolled my full 408. So at the fourth interim, I force a go-no-go decision, and I've set the success futility threshold using the same Bayesian posterior probability at 0.92. Now, one of the problems in a phase 2 trial of stopping early is it can impair our ability to correctly pick a dose. So to protect my dose selection, I add an additional success criteria, which is I want to see the dose with the highest probability that it has the maximum response. I want that probability to be 0.5 or better. And as there are 3 arms, so if one has the probability of maximum being better than 0.5, it's quite likely that the other arms will have a considerably less probability, and we're probably done in choosing our arm. It's kind of becomes unlikely that probability will shift. Unless we had a situation where one arm was very bad and 2 arms were pretty much the same. Things I didn't look at, we could have perhaps tried an even earlier interim when there was only about 50 subjects complete. I could have used a different end of phase 2 threshold. I used 0.92. It looks like which slightly over controls the type 1 error. It's less than 0.2. Possibly had more frequent interims. And I could also probably try tuning the PR max threshold. As 0.5 gave reasonable operating characteristics, I've just gone with it. Obviously, it means that I don't need that threshold to be higher. perhaps I could get away with it being a little bit lower and still be choosing the right arm the same amount of time and maybe stopping a bit earlier. And then, the adaptive allocation. So I use adaptive allocation, and I'm allocating to simply preferentially allocating to those with the highest probability of being the max. I've used information weighting, so it takes into account how many subjects are on the arm, down weights the probability of allocating to it, the more subjects are on it. And I've also allowed control to be allocated to adaptively. What FACS does there is it gives control equal probability of being allocated to as the treatment arm with the highest probability of being allocated to. So it gets us close to equal allocation to control and the best arm, and so maximizes our power. The interims, so my first interim is at 252 enrolled and every 8 weeks. And then this is the setup where there's no early stopping, so that I could do that. Oh, I got it open. I could do this graph, because of course, if you put in early stopping, then there won't be the data as to what the simulation would have been at the end. So I use these no early stopping simulations to get this graph to choose the criteria. Okay, so that was just looking at what the adaptive allocation rule was. And hopefully that's followed most of Kurt's advice in the previous talk. So compared to the fixed design, let's have a look. So the overall success rate in the null is 0.19, so it is kept below 0.2. The power is, in the different scenarios, 0.91, 0.93, 0.94, so that's good. The probability of selecting the correct arm is 0.90, 0.76, 0.73. That's slightly reduced. If you can see the original down here. So perhaps there isn't any extra tuning to do with the PR max stopping criteria. Compared to the fixed design, we've got an estimated sample size of around 320, expected sample size of 325 to 330. So you can see that sample size increase. But the duration is smaller. In the alternate, it's 54 to 55 weeks compared to 68 for the fixed. And this is because we're not waiting till our 252 are complete before doing our analysis and making our decision. And of course, what we're hoping for now is that by combining the phase 2 data into phase 3, because our phase 2 is bigger, our phase 3 can be a bit smaller. And we opt to reduce it to about 60 subjects per arm in phase 3. But what we do is, instead of specifying that the phase 3 size is a total of 120 subjects, we just say the overall cap is 408 plus 120, 528. So if phase 2 transitions early, actually we will be able to have more phase 3 patients. But we're also going to add interims to allow the phase 3 to stop early. And that's to do the interims. We're going to do this thing that we mentioned in the group sequential, but didn't cover, which is a Bayesian, what we call a Bayesian Goldilocks design, where instead of using group sequential boundaries, we use Bayesian predictive probability boundaries. And these work a lot better in these situations where there's a long time to the endpoint. which is a situation typically not covered well in the group sequential design. So just so very quickly, just to illustrate this predictive probability idea, imagine we've got a simple trial, single arm, 100 patients, and we want to show that the probability of success is greater than 0.5, and we want a confidence of 0.95 or greater, but the probability of success is greater than 0.5. If we do a simple beta prior, one prior, and then a simple beta binomial analysis, if we had 58 subjects out of 100 successful, we would only have 0.944 confidence that the probability is greater than 0.5. If we had 59 successes out of 100, we'd have a probability of 0.96, so we'd be over our required 0.95 threshold. So we need to see out of our 100 patients, 59 successes to be successful. Now, let's say we look at the data halfway through, and we have 28 out of 50 subjects successful. So the success rate is 56%. So that's great. We're over the 0.5, but what's the probability that we will see our 59 successes at the end of the trial? And unfortunately, that means we're actually going to have to see a greater number of successes. We need 31 out of the next 50 subjects, 62% rate in the second half of the trial. And given what we've seen so far, what's the probability that we will actually see 31 subjects? Well, we can do a simple binomial. If we're seeing 50 subjects and we currently believe the true rate is 0.56, what is the probability we will see another 31 subjects? And it turns out it's about 0.24. And so our probability of success in this trial is now 0.24, and our probability of failure is about 0.76. So very quickly, hopefully that you see how predictive probabilities work and how we use them is we're going to look at the data at the interim and predict our probability of success of a p-value test. For instance, probably the p-value is less than 0.02 if we were to continue to the end of the trial. And if that probability is too low, say less than 10%, we could say, well, the odds of success are now too poor, and we will stop the trial for futility. Alternatively, if we say, well, given what we've got now and the subjects we've recruited up to this point, what's the probability if we just follow them up, that will be successful? We'll have a final p-value less than 0.02. And if that probability is sufficiently high, say greater than 99%, then we can stop enrolling and we'll just complete the follow-up of our existing subjects and have an extremely good chance that the trial will be successful. This does result in a very small inflation of type 1 error, and the final alpha value needs adjusting to compensate for it, which we do by simulation. So we're going to use that in our phase 3. And we're going to set up a small number of interims in the phase 3 based on the number of subjects we have complete. So we'll have the first interim at 150 complete, which we might have already, only counting the arm that we've taken to phase 3 and the control arm. And of course, at that point, there will be a further 156 subjects that will have been enrolled and not complete. And then we'll do interims every 50 subjects completing. And again, we simulate without any early stopping bound set, and we use that nice final versus interim scatter plot to check the boundaries, which I don't have time to show you. And what that comes down to is some nice conservative boundaries are we only stop for success or stop enrolling for success at the first interim if our probability of success is better than 0.99, or subsequent interims better than 0.98. And for all interims, we stop for futility if the success at full enrollment is less than 10%. So with this 26 weeks to the final endpoint and simulating an accrual of 6 patients per week and assuming a 26 weeks gap between phase 2 and phase 3, using the separate phases, we have this overall time and error of 0.0035, this power of 80% or a little bit less, our probability of selecting the right arm of 80% down to 70%. The maximum sample size is 422 across both stages, with an expected sample size of 277 in the null and about 400 to 410 in the alternates, and a duration of 80 weeks in the null and 140 weeks in the alternate. Using this seamless phase 2, phase 3, retaining the data from phase 2 into phase 3, being adaptive in the phase 2, and having early stopping in the phase 3, the power is now 90% in the alternates, so considerably improved. The probability of success with the right arm is better in one case and the same in the others. The maximum sample size is increased. Under the null, it's quite considerably bigger. It's 400. Under the alternates, it's actually only a little bit bigger, 440, so only about 30 subjects bigger. And the duration has come way down. It's about the same in the null, but in the alternates, it's now below 100 weeks. So we've saved nearly a year, over 40 weeks. And we've got the better power. So as we reach the end, thank you for attending. As normal, a link to the recording will be sent out. But at the end of the series, we'll put all the links to the recordings on the website along with copies of the slides. If you've got any questions, please just email us, myself, Tom at berryconsultants.com, or Kurt at berryconsultants.com, or the whole fax team at fax@berryconsultants.com. And of course, if you're interested in a detailed demo of fax or a free evaluation of copy of fax, then just ask us. Thank you very much.